Good evening, everyone. Good evening and Jai Hind. I hope I am properly audible and visible to all of you. Good evening, Shivanand. Good evening, everyone. I hope I am properly audible and visible to all of you. Good evening, Prachi. Good evening. Okay, so I guess I'm properly audible and visible to all of you. Thank you. Good evening. Okay, so last day I have asked you this one question, that when we are talking about the food risks, like India, the Indians, the choices of food by Indians and the risks which are associated with this. So with respect to that, I have got many of the answers from you. One is from, I think, from Anjali, that is food adulteration, overconsumption of oily and fried foods, excess salt and food, sugar, fast food, etc. Then there is from Sonu also, because cultural preferences, okay, street food hygiene, overuse of spices and salt, childhood malnutrition, okay. Then there is from AD Motify, okay, so that is obesity and diabetes, malnutrition and nutritional deficiencies. Then you have also mentioned about steps which are needed for this. Then I have got one answer from Shaheen also, that what are the risks due to food choices of Indian. I have mentioned 15 different choices, 15 different parts of it, that from obesity, diabetes to liver problems, kidney problem, micronutrient deficiency, micronutritional deficiency, high blood pressure. Yes, that is also there. Then is from Lucky also, that non-communicable diseases are rising, steps which are needed, that is also mentioned. Okay. So I think many of you are giving the answer to that question. And see, this is one of the very important part of your GS paper 2 also. Like the health and nutrition, that remains one important part. We are having this double burden of malnutrition. So that is the first thing, some of the answers which have been given by you. One answer is from Sahil also, that is food poisoning, obesity, more non-communicable disease. So these were the answers which has been given. Okay. 2025, nahi, 2025 वाले students, just focus on your revision now. Okay. There is one question, sir, न्यूज़पेपर की जगह मैगजीन पढ़ने से एग्जाम में कोई रुकावट नहीं होगी. देखो, today also and many a times I have discussed about this. See, there is one problem, like which many of you are going to face, that is framing sentences in the examination. You will have this issue of framing sentences in the examination. Why? Because when you are reading the newspapers, you get to know that how the things are written. But when you are not writing, not like going through those newspapers, if you are simply relying on magazines or books or class notes, then you will not learn that art of writing long sentences or explaining something in your own words. So if you have to write, you have to read. There is one simple concept. If you have to write, you have to read. And in newspapers, they are giving it in raw format. In magazines and class notes, we are giving you in a very simple manner. So even if you are reading one single article per day, that is also sufficient, but you have to read at least one article per day from the newspaper. So that is always required. Okay. Yes. So let's start today's topic. See, the things which we are going to see in today's session, one is H5N1. The first topic which we are going to see is H5N1 and genome sharing. That is the first thing which we are going to discuss. Second thing is all about NIA, the National Investigation Agency, how it is different. Right now it is there in the news also. So we are going to discuss about this. Then we are going to discuss about the semi-cryogenic temperature and engine. temperature, technology and engine. Then we are going to talk about RNA-based antiviral. We are going to talk about this one, United Nations Security Council, which has condemned this Pahalgam attacks and has said that those who are behind this, they must be brought to justice. Okay. And then we are going to talk about cheetahs also. So these are the topics which we are going to see in today's session. And before we go into that, we will see also some practice questions. We are also going to see some practice question. Okay. Now, before we go into that part, there is one information for you. Those who are starting for 2026 or 27 or later, we are starting with the foundation batches for 2026. We are starting with this foundation batch for 2026, and you are also getting a free prelims test series for 2026 with this. So it is there in English medium also. It is going to be online. So you can visit the website. You can visit KGS website, and from there you can enroll in it. Okay. Or you can go to KGS app also, and there you will find the option to enroll into this. Okay. So this is one thing, foundation batch for 2026 is going on. You can enroll in that. Okay. Now, coming to the question. Last day we have discussed about one thing, POXO. In light of that, we have this question. Which of the following statement about the Protection of Children from Sexual Offences Act 2012 is are correct? It defines a children as a girl below the age of 18 years. The act mandates that the child's statement be recorded in presence of child's parent or a trusted adult. The act mandates the reporting of a crime even if it has been done by consensus among juveniles. So this is one part. Which of the following is are correct with respect to Protection of Children from Sexual Offences Act 2012? Economic survey कैसे पढ़े 2025 के लिए? Priya, see, on KGS channel, YouTube channel itself, you will find that economic survey has been discussed. So I will suggest that go through that economic survey. From there you will get the entire idea. Or otherwise, if you want to do it from your own side, just go through the important charts or tables which has been given in economic survey. There are around 20-30 important charts or graphs which are made in economic survey. If you just go through that, it will be sufficient. Then Anjali, of course, your comment is also visible. I am quite good. I hope you are also good. So what will be the answer for this? B option. B option is 2 and 3. B option, B option, C option. See, this is one topic which we have discussed in the previous session on Friday, prevention of children from sexual offences. Okay. And I think most of the people are going for answer B. That is correct also. Basically, it is not defining the girl only. It is not simply saying that a child as a girl only. It is defining a child as a person. So it is independent of gender. It defines the child as a person below the age of 18 years. Okay. So this first option is incorrect. It defines a child as a person below the age of 18 years. Second option is correct, and third option is also correct. So we have seen all these provisions in the last session. Answer here will be option number B. Okay. Coming to the next question. Consider the following statements regarding the World Trade Organization. It was established under the Marrakesh Agreement in 1995. The Appellate Tribunal is the first level dispute resolution mechanism of WTO. It deals only with trade in services, trade in goods, and not in services. It deals only with trade in goods and not in services. So this is question number 2. This is question number 2. What will be correct here? last question was EG1. Answer was not D, it was B. What should be the answer here? Question number 2. Consider the following statement regarding World Trade Organization. Okay, one answer from David as option number A. Anyone else? Is this a tough question? WTO we have discussed on the last day. We have seen what is WTO. We have seen about its relevance, whether it is relevant or not. So what will be correct? A option, C option, A option, A option. Now, if we look at this question, the first one is saying it was established under the Marrakesh Agreement in 1995. That is correct. Second is saying the appellate tribunal is the first level dispute resolution mechanism of WTO. That is incorrect. First, there is setup of a dispute resolution board. Okay. Then after that, this is the appellate tribunal. So if it is written appellate tribunal, it already means that after this, after non-settlement of dispute at the dispute settlement board, then it is applied, or then it is again appealed at this appellate tribunal. So second option is incorrect. It is not the first level dispute resolution body. It deals with trade in goods and not in services. So that is also incorrect. So answer here is option number C. That is one only. Coming to the third question, with reference to antibiotics, consider the following statements. It helps us to fight against bacterial or viral infections. The overuse of antibiotics can lead to antimicrobial resistance. Third is saying one of the main reasons behind AMR is people's genetic predisposition. Which of the statement given above is are correct? Last one option is D option 2 and 3. If it is not visible, I am writing it for you. 2 and 3 only. 1 and 2 only, that is option A. B is 1 only, C is 2 only, and D is 2 and 3 only. So question number 3. Question number 3 about antibiotics. First one is saying it helps to fight against bacterial or viral infection. The overuse of antibiotics can lead to antimicrobial resistance. And one of the main reasons behind AMR is people's genetic predisposition. So what will be the answer? Okay. A option, D option. So tough luck today. Antibiotics help to fight against bacterial infection, not the viral. Viral infections are not treated by antibiotics. So first option is incorrect. The overuse of antibiotics can lead to antimicrobial resistance. That is also correct. One of the main reasons behind AMR is people's genetic predisposition. This is incorrect. It has been asked even in UPSC exam. So answer here is option number C. That is 2 only. Okay. So answer here will be option number C. So in this question, tough luck. Okay. Antibiotics are not for viral infections. Antibiotics are for bacterial infection. Okay. For viral infection, there is no as such medicine which can help to remove it. But generally there are antivirals which are given for controlling the symptoms, not for treating them. Okay. So answer here is option number C. That is 2 only. Okay. Now coming to this topic of today, the first one is H5N1. Okay. Now there are certain things which you have to see here. One thing is there is one thing called as H5N1. So we have to understand what is this H5N1. Second thing is we have to also understand what is genome. And third thing which we are going to discuss is GIS aid. So there are three things which we are going to learn under this article. One is the H5N1, second thing is genome, and third thing is GIS aid. Okay. Now you all know that this H5N1. Now before we go into that, let me add the context here. There is a Pune based National Institute of Virology, NIV. So NIV, that is National Institute of Virology. So it was not sharing the data with one of the global organization. That global organization is Global Initiative on Sharing All Influenza Data. It is All Influenza Data, Global Initiative on Sharing All Influenza Data. So there is a mechanism which says that all the labs of the world, they have to share the data as part of this GIS aid program. India is also a member of it. So India has to also give the data. But the Pune Institute of Virology, National Institute of Virology, was not sharing the data. So there was a report about this in the Hindu itself. And after that, there has been reporting of this by the NIV. Okay. So first thing is, we will try to understand what is this H5N1. That what exactly is this H5N1? It is basically a strain of bird flu. It is a strain. A strain means variant of bird flu. And what was the data which we are talking about? Basically, the data was that a 2-year-old girl from Narasaropeta in Palnadu district of Andhra Pradesh, she was having this H5N1 sequence. A 2-year girl from Andhra Pradesh, she has got this H5N1, and this was confirmed, but the data was not shared. So then there was a newspaper report about this. After that, there has been sharing of this data. Now, if we talk about the H5N1, you can see there are 4 different strains of this avian bird flu or avian influenza. There are 4 different strains of avian influenza. H5N1, it is in circulation since 1997. H7N9, 2013. Then we have H5N6, 2014, and H5N8 since 2016. Now, out of all these 4, this one is the most deadly one. It is the most deadly. It is the most deadly one. Why? It has killed more than 40% of the farm cattle which it has infected, farm birds which it has infected. Okay. So this avian influenza, it has infected huge number of birds and 40% of them has died. Not only this, it has also infected by wild birds and poultries. Okay. Then at the same time, now we see that it is also impacting some other mammals as well. It is impacting other mammals as well. Okay. So this is one of the deadly virus, avian influenza, which is going on. Okay. This has caused huge outbreak in 1996 also, and throughout the world, it is causing problem. Okay. And it can infect humans also, but rarely. Okay. Can infect humans, but rare cases. But there is rare cases. Like you can see, in India, when there is a reporting from Andhra Pradesh that a 2-year-old girl is having avian influenza, so from there you can understand that how rare it is. It is not common for humans to get this avian influenza, but when it gets there, then it becomes an important news article. Okay. So this is one part. Now, there is second thing that we are sharing genome data. So I think all of you can tell me what is this genome data. Can you tell me what is this genome data? Can you tell me this part? What is this genome data? What is this genome data? Anyone? or genome sequencing. What do we mean by this? What is genome data? And why this is crucial? Like, what is the application of it? What is genome sequencing or genome data, and why this is crucial? First thing is, what is this data all about? It is information about sets of DNA genes, okay? Fine. It is basically complete set of, it is basically complete set of DNA, RNA, genes, etc. So this is a complete set of DNA, RNA, genes, etc. That is our genome. Okay. All the proteins, the coding, non-coding proteins, it's and everything. Whatever genetic material is there, okay, all the genetic material, all genetic material. That is basically the genome of any organism. Okay. And what is the application? Like, how it is useful? How it is useful if we talk about this? The application of it. So this genome data can help in diagnosing the diseases. Diagnosis of diseases. Like, suppose if in any of the population a genetic disease or a particular disease is more common, so we can try to understand that what causes this, what is the reason behind this. Okay. It can be used to study viruses or bacteria, or to study microbes or viruses, etc. It can also be used for developing personalized medicine. For developing personalized medicine. It can be used for developing personalized medicine. We can also use this for agricultural improvement or evolutionary studies. Okay. Agri reforms, agri improvement. Okay. So these are all different things. Even for new drug, okay, new drug development, we can also use this. Okay. So these are the probable areas where we can use this genome sequencing. Now, the third important thing is, what is the GS Aid? So GS Aid is basically a global platform that promotes the rapid sharing of data on influenza viruses and coronaviruses. Okay. So this is a global platform which allows the sharing of data with respect to avian influenza and even now coronavirus. Okay. So what we see that it was launched in 2008, and why especially? Because of sharing of influenza virus data. Okay. So H5N1 was the special region why this was started. So it is managed by the GSAD initiative, which is a non-profit organization based in Germany. Okay. So that is the role of it. GS Aid, it is sharing the data to different people. What type of data is being shared here? Like, what is the data which people are sharing here? If you look at this, viral genome sequences, metadata like location, date of sample collection, etc. And even information about the mutation and variants. That what all variants are there, what all strains are there, that is all being shared in this GS Aid global initiative on sharing of avian influenza data. Okay. Now, what we see that the data contributors, they retain the right over their data. Okay. It is open database, means people can see it, but if they are using it, they have to give the credit. Okay. So what we see that access to data is free, but users must register and agree to respect usage condition. Okay. And also encourages transparency. So this is one part, that GS Aid, genome sequencing, and the H5N1. These all three things were in the news which we can take from this one article. Okay. Now, the second article is about this NIA. Okay. Genome is related to a structure of living organisms and their work. Recently, you may have seen this, that a new animal, there is a claim by one of the company that they have resurrected an animal which is like, which was extinct thousands of years ago, which was extinct something around like 13,000 years ago, that has been resurrected. So you may have seen that news. That an animal has been brought back to life using the genome data. So that is also possible thing. Like we can have conviction and all. Okay. We can have, sorry, cloning and all possible through this genome and everything. Okay. Now there is this one organization, NIA. So tell me, what do you know about this organization? Like how it is different? How this force is different? What are the powers which it is getting? What are the powers which are given to this? How is it different? Dire wolf. Dire wolf is there. Yes. Dire wolf has been resurrected. And people are saying that other animals which have become extinct, they might also be done in the same manner. Okay. So tell me about NIA. Right now this was there in the news. So it is the Federal Counter-Terrorism Agency. Now, if we look at its like power, like if we are talking about the scope of its power, what is the jurisdiction of it? Okay. This is of course a statutory body. It was formed after the 2008 Mumbai attacks. After the 2008 Mumbai attacks, we have got this NIA Act, and from that itself, this agency has been created, a federal agency. If we talk about jurisdiction, its jurisdiction is entire India. And any world means it has this jurisdiction in all the parts of India, in all the states of India, in all the union territories of India, plus even beyond our border. If there is a case, let us say in Canada, and we have an agreement with Canada, then this NIA can also investigate cases in Canada. Okay. So it has been given the power to investigate beyond India also. So it is not limited to the borders of India, it can go even beyond that. Okay. If we talk about this, it has extensive power. In case of terrorism cases, it can register a case, arrest people without the permission of state government. Okay. So what we see that no permission required from state government. So it is not as the Central Bureau of Investigation. No permission required from state government. It is a federal agency, so it has jurisdiction over all the parts of India. It is not like CBI that you can withdraw the general consent, you can give a special consent. It doesn't require the consent. By default, it has been given consent over all the parts of India. Okay. If a state want, they can ask the NIA to start an investigation. But if center wants, they can have this investigation with or without the permission of the state. So here the state permission doesn't matter in case of NIA. It is a federal agency. Okay. Now, what are the laws? What are the laws where it can take investigation? The law is Atomic Energy Act of 1962, UAPA, Unlawful Activities Prevention Act 1967, human trafficking, offenses related to counterfeit currency or banknotes, manufacture or sale of prohibited arms, cyber terrorism, or offenses under the Explosive Substances Act 1908. Earlier, its power was limited to these two things, atomic energy and unlawful activities. But later on in 2019, these 5 things were also added. So as per the latest amendment, it has power over all these laws. So in all these cases, it can investigate the crime in each and every part of India. Okay. And can you tell me, what is the conviction rate? What is the conviction rate? Means if it is arresting, or if it has got 100 cases, if it has got 100 cases, so what is the conviction rate of NIA? What is the conviction rate of NIA? Can you guess? Ashutosh, I will take that. Don't worry about it. I will take that. Okay. So tell me about this part. Conviction rate, if we are talking about this, can you guess what will be the conviction rate of NIA? How much is the conviction rate? Any guess? How much conviction it is having? Take a guess. Like normally you see that police is having a conviction rate of 10%, 15%. Some of are having even lower than that, 5%, 6%. This has got a conviction rate of 94.7%. It has got a conviction rate of 94.7%. Okay. So that is very high conviction rate. 94.7%. Okay. So it is like enormously successful organization in this term, that if it is having 100 cases, out of that 94% or more than that, they are ending up in jail or they are convicted of a crime. So that is the conviction rate of NIA. Okay. So it is very high in that terms. Okay. Now let's see some questions. Let's see some questions. Like we have discussed 2 topics. Let's see some questions on the basis of it. The first one is here. Aerial metagenomics best refer to which one of the following situation? Collecting DNA sample from air in a habitat at one go, understanding the genetic makeup of avian species of a habitat, using airborne devices to collect blood samples from moving animals, and sending drones to inaccessible areas to collect plant and animal samples from land surfaces and water bodies. So this is a previous year question also, and I think many of you have read this. I have also taught it in the crash course program. So what will be the answer here? Aerial metagenomics. What does it refer to? Aerial metagenomics. It refers to what? Collecting DNA samples from air in habitat at one go, understanding the genetic makeup of avian species of a habitat, using airborne devices to collect blood samples from moving animals, and sending drones to inaccessible areas to collect plants and animal sample from land surfaces and water bodies. Okay. So what will be the answer? Okay, I think 3 answers as option number A. What about other people? What will be the answer here? Yes. So I think you know that It is about collecting DNA samples from air in a habitat at one go. Basically, it is the sample of all the microbes which are there. Okay. All the microbes which are there. When we are connecting their, when they are collecting their DNA samples, okay. So that is what we see, that is called as metagenomics, aerial metagenomics. Okay. So metagenomics is collection of the DNA sample, and aerial means the one which is present in the air. Now, this is one question: low temperatures or cryogenics find application in. We are going to see this topic, but before we go to that topic, there is this one question: low temperatures or cryogenics find application in space travel, surgery, and magnetic levitation. Surgery, magnetic levitation, and telemetry. Space travel, surgery, and telemetry. Space travel, magnetic levitation, and telemetry. So where do we get this? Where do we use this? Low temperatures or cryogenics. This is question number 5. This is one of the previous question which was asked way back, like 20 years ago. This was the question, that cryogenics, where do we have the application of the cryogenics? Where do we use them? One option is space travel, second option is the surgery and magnetic levitation. Okay. Second is saying like surgery, magnetic levitation, and telemetry. Third is saying space travel, surgery, and telemetry, and space travel, magnetic levitation, and telemetry. Okay. And so this is this is one thing which you may not have read yet. Like space travel, I think every one of you know. In space travel we use it. In surgery also we use it, and even in magnetic levitation we can use this cryogenic. Okay. Where we are making the electromagnets, we can use this magnetic levitation. Telemetry, just like sending of the information over long distances. Okay. So that doesn't require this cryogenics. So telemetry, in that case we don't require it. Okay. Like exchanging the signals, exchanging informations or others over long distances. So there we don't require it. But in all other three things, space travel, surgery, and magnetic levitation, we require this cryogenic technology. Okay. Now, we have this one thing, semi-cryogenic technology. That is the recent thing which we have to see. There is one thing called as semi-cryogenic technology. Cryogenic. So see, before we understand the semi-cryogenic, we also have to understand that what is the cryogenic technology. Okay. In case of cryogenic, when we are talking about the cryogenic, there are cryogenic engines, and in this we are using liquid oxygen. This is called as liquid oxygen and liquid hydrogen. We are using liquid oxygen and liquid hydrogen as the fuel. Okay. So this is liquid hydrogen. Now this is the fuel and this is the oxidizer. And cryogenic means they are all kept at like very low temperature, like -173. Okay. They are kept at low temperature like -170 degree or even more than that. So that is our cryogenic engine. Okay. So in cryogenic engine we are having it at a very low temperature. We are having the oxidizer as well as fuel, at very low temperature. So this is one part. But in case of semi-cryogenic, what is the difference here? When we are talking about the semi-cryogenic engine, so here we are still using liquid oxygen. We are using liquid oxygen. and this one is stored at cryogenic temperature. This one is stored at cryogenic temperature. Then we are using kerosene as the fuel. We are using kerosene. Okay. We are using the kerosene as the fuel. And this kerosene is also like a refined form of this. This kerosene is like a refined form, and we are using it as the fuel. We are using it as the fuel. But here at normal temperature. This one is stored at normal temperature. So that's why this engine will be called as semi-cryogenic engine. Here we are storing the kerosene at normal temperature and this oxygen at cryogenic temperature. So that's why it is called as semi-cryogenic engine. So we are replacing hydrogen by kerosene. So that is what we see, that it is a semi-cryogenic engine. Why we are doing this? What is the advantage here? The advantage is this: cheaper. The refined kerosene is cheaper than the liquid hydrogen. It is easier to store also and transport also. It provides a high amount of thrust also, and it can be used for the reusable launch vehicles. It can also be used for reusable launch vehicles. So for these reasons, now India is also developing this semi-cryogenic engine. We are trying to develop this semi-cryogenic. Okay. Our cryogenic engine, which launch vehicle of India is using cryogenic? एप्लीकेशन लिखने में किस चीज के एप्लीकेशन लिखने में, किस चीज के एप्लीकेशन लिखने में गलती हुई है? And tell me, which vehicle of India is using cryogenic engine? Which vehicle of India is using this cryogenic engine? Medical में? I'm not sure what you are asking about. Okay. Now tell me this one part, which vehicle of India uses cryogenic? Uses or use cryogenic. Which vehicles of India are using this cryogenic? LVM is there. LVM 3 launch vehicle Mark 3 is there. So yes, launch vehicle, if we talk about the GSLV or LVM, so there we are using this cryogenic engine, correct? Okay. So we are using this GSLV where we are having this cryogenic engine. So this is one part in semi-cryogenic technology. We are going to use this in the next generation launch vehicle, NGLV. In the next generation launch vehicle, we might be using this semi-cryogenic. Okay. So that is the aim here. Okay. Now, coming to the next one article, this is also a very important article, RNA-based antiviral. So antiviral spray. There is an antiviral spray which has been developed. An antiviral spray. Okay. Normally, if we talk about this, suppose if you are looking at this cucumber, you will find that a huge amount of the plants, they are getting destroyed due to viruses attack. Okay. Around 40% of the crops across the world, they are affected by viruses. In India also, what we say that there is loss of 25 to 30% of the crop yield due to various viruses. Okay. So what we see that globally 40% crops affected by virus. India also, if you look at this, if we talk about India also, what we see that 25 to 30% yield loss. So in this context, like when we are using antibiotics, when we are using the antibiotics or insecticide or pesticide, they can be used to treat the living organisms. They can be used to kill the living organism. But viruses are not living. So if viruses are attacking our plants, then we have no rescue. We have no other option. So when you see the cucumber like these in the market, I think many of you may have eaten this also. You may have seen that in market, there are various cucumbers, they are having some dots on it. You may have got it at various other places. You may have got it in the market. These cucumbers are generally impacted by virus. Okay. Now, you don't have to worry about it. You don't have to worry about it. The plant-based viruses do not impact the humans easily. If it is a plant-based virus, they do not cause harm to the humans easily. Okay. If there is an animal-based virus, they impact us. Okay. So, keeping this in mind, there has been development of an antiviral spray. There is a development of antiviral spray. And what it does, it introduces, this one is going to introduce a double-strand RNA in the genome of plants. In genome of plants. This one is going to activate the immune system. It is going to activate the immune system. And what will be the impact? Plant will fight back against viral infection. Plant will fight back against viral infection. So this is one thing which we see, that there is an antiviral spray which has been developed, and this antiviral spray is going to be sprayed on these crops. They will introduce a double-stranded RNA in the genes of the plants or in the genome of the plant. This RNA is going to activate the immune system of the plant. Following that, the plant is going to kill the viruses or going to fight with the virus. Okay. So this is an antiviral spray which has been introduced. Now tell me about this plant. Tell me about this part. Will it introduce a genetic modification in the plants or not? Will it cause genetic modification here? Is it possible that this antiviral spray will this cause genetic modification? Will this cause genetic modification or gene mutation? Or gene mutation? So see, normally we understand that this may cause some genetic mutation, but see this part, it is just a RNA. It is only a RNA, so it will help in translation of protein. It will help in translation of protein, but no change in DNA. But no change in DNA. And this will also naturally gets degraded. Naturally, it gets degraded. Okay. So, in fact, there is no gene mutation. No gene mutation. So, what we see that on one hand, it is helping to fight back. It is helping to fight back against the viruses. On the other hand, it is also not causing any genetic modification. So this gives a lot of opportunity. In many of the plants, we are trying to have genetic modification. We are trying to do genetic modification in many of the plants. Why? Because we want to fight back against some of the bacteria or viruses. Now, if we are looking at this, an antiviral spray without causing any change to the DNA can also help in getting that response. Okay. So it can also help to get that response. So it gives us an opportunity that in future we may look at this option to make our plants much more better, much more capable of fighting against the infection, while at the same time we don't even need the genetic modification of them. So even without genetic modification, we can fight back, or we can develop a significant fight back mechanism in the plants. Okay. So this is the antiviral spray. Okay. So this is the antiviral spray, which is RNA based antiviral. I hope this part is clear. What is antiviral spray? Now, so there is this one article, United Nations Security Council. Okay, so there is just this one information that it has said that bring the Pahalgam attack sponsors to justice. Okay, you know that when we are talking about the United Nations Security Council, UNSC, it is a permanent body. It is one of the principal organ. It is one of the principal organ of UN. Okay. You know there are some principal organs, some agencies. I think you know that. Okay. So this is one of the principal organ of United Nations Security Council. It has got P5 members, permanent 5 members, plus 10 members who are elected. Okay. Plus 10 elected members. If we talk about decision making, decision making happens by 9 by 15 votes. Okay. It happens by 9 by 15 votes. But P5, they can veto any decision. They can veto any decision. Okay. So this is one part. What we see that it takes decision by 9-15. But in this situation also, what we see that they require, like any P5 member, they can veto any decision. So right now they have come in support of this one part, that they have said that bring the Pahalgam attack sponsors to justice. Okay. So this is one part, like everyone is condemning the terror strike. Okay. So this is one thing which has been done in case of Pahalgam case right now. Okay. If any further things has been done, we have already discussed about this Indus Water Treaty. We have seen about persona non grata and other related things. So if any new development comes, we will see that also. Okay. There is one more thing. There is one more thing which was there in the news. Basically, what we see that in Kuno National Park, we have seen that a cheetah has given birth to 5 cubs. Okay. It has given birth to 5 cubs in this Kuno National Park. We have recently seen that there is one second home. There is a second home to cheetah. Can you tell me which is the second home to cheetah in India? Which is the second home to cheetah in India? Last week we have discussed about this. There is a second home of cheetah in India, and there is a breeding center also. There is a breeding center also, which is planned at Bunny Grassland. This has been planned at Bunny Grassland. Okay. So there is a second home of cheetah. What is that? What is the name of that location? The MP is there. What is the name of the park? What is the name of the area where we have planned for the second home? Where have we planned this second home of the cheetahs? Last week we have discussed about this. There is a special area in Madhya Pradesh itself. That is Gandhi Sagar Wildlife Sanctuary. Okay. So here we see that there is Gandhi Sagar Wildlife Sanctuary. So that is also there in Madhya Pradesh itself, Gandhi Sagar Wildlife Sanctuary, and there we see that cheetah are going to be reintroduced in this area. Okay. So this is one more information. Now look at this one question. Consider the following: black-necked crane, cheetah, flying squirrel, and a snow leopard. Which of the above are naturally found in India? 1, 2, and 3 only, 1, 3, and 4 only, 2 and 4 only, and 1, 2, 3, and 4. Now you have to give answer as per 2025. Try to give answer as per 2025. Which of them are naturally found in India? First one is this black-necked crane, cheetah, flying squirrel, and then we have this snow leopard. So which of the above are naturally found in India? Which of them are naturally found in India? Yes, three answers as B option, 1, 3, and 4. Cheetah is not naturally found in India. What we see that they have been introduced. So they are not naturally found. Now they are not naturally found. They have been introduced in India. So still the answer will be same. They are not Asiatic cheetah. They are African cheetah which has been introduced in India. Okay. So right now we can say that only 3 of them, they are naturally found in India. Black-necked crane. Then we have the flying squirrel. Then we also have the snow leopard. Okay. One last question. The Security Council of United Nations consists of 5 permanent members, and the remaining 10 members are elected by the General Assembly for a term of 1 year, 2 years, 3 years, or 5 years. The answer will be option number B. So look at this question, question number 7. The Security Council of United Nations consists of 5 permanent members, and the remaining 10 members are elected by General Assembly for a term of how many years? Yes. So I think there you know the answer. Yes. It is done for a period of 2 years. So here that is correct also. In question number 7, the answer is B. So this is one question for you. How genome sequencing is going to help India? Mention some recent steps related to genome sequencing in India. So these are the 2 questions which you have to answer. First thing is how genome sequencing is going to help India. Second is mention some recent steps related to genome sequencing in India. Okay. So mention your answer in the comment box, and the PDF of this session is going to be there in the Telegram channel. Okay. So that will be all. One more information for you. The current affair magazine of this April month is available on this store of Khan Global Studies. So you can scan this QR code also, or you can visit the website of Khan Global Studies. Okay. So from there you can get this current affair magazine of April 2025. Okay. So it is there in Hindi also, English also. And that will be all for today. Just answer that genome sequencing question. There we will meet tomorrow again at the same time, 5 p.m. So give the answers of the questions and then we will see the other topics. Okay. Thank you everyone. Take care and all the best. Thank you.